Hey everyone, my name is Jessica Dela Cruz. Today's date is February 5th, 2019. It is week 5 lab for biochemical testing or microbial identification. It is a carbohydrate fermentation lab. So in this lab, I am going to be incubating S. cerevisiae and S. epidermidis cultures in fructose, glucose, and mannitol broths to categorize each microbe based on metabolic properties. Okay, so here I've already done the my five fennel red broths, and I insert the germ tube in there with the corresponding glucose, and then I did the fructose as well. Okay. All right. And then so let's do this. So now I'm going to grab my last vial. It is number 3 of the fennel red broth. I'm going to do the mannitol for S. epidermidis. I'm going to grab this. So I'm going to open the tube and I'm going to flame the lip to sterilize. Okay. And I'm going to grab the mannitol that I got from the lab kit, and I'm going to pour the remaining of this, the other half, because I already did the first half. Okay, just put that in there. Okay, right, and I'm going to flame the lid again to sterilize, and I'm going to close it. I'm going to shake it so that the powder dissolves. Okay. Now I'm going to get my germ tube. I'm going to open it up. I'm going to flame the lip again to sterilize. And I'm going to grab my germ tube that I've soaked in alcohol, and I'm going to place the germ tube into the fennel broth base. I'm going to place the germ tube so that the opening of the germ tube faces towards the bottom of the fennel red broth. Okay, so just like that. Okay, so it's going to look like that. Right, then I'm going to grab my S. epidermidis active culture. I'm going to open this. Okay. Okay. And I'm going to flame this. Okay. I'm going to grab my pipette and just shake to dry, because it was sterilizing in alcohol. Okay. So I'm going to grab some of the active culture of the S. epidermidis. Okay. And I'm going to add 4 drops into my fennel red broth base. 1, 2, 3, and 4. Perfect. Then I'm going to dispose of this into my 10% bleach solution because I am done with it. I'm going to flame this, my active culture. Put the cap back on. Alright, and I'm also going to do this to my fennel red broth. I'm going to cap that. Alright. Okay. So the reason why we do the carbohydrate testing is it determines the product of the fermentation and thus may be used to differentiate among groups or species of microbes. So in these tests, microbes are cultured in a series of broths, each containing an isolated carbohydrate such as glucose, fructose, or mannitol. The acid and products of fermentation lower the pH of the broths. So phenol red is commonly used as a pH indicator in carbohydrate fermentation tests. Phenol red turns yellow in acidic solutions. The germ tubes that is placed in each one of these are used to detect the carbon dioxide and hydrogen gases that may be produced by fermentation. Also, these small glass tubes trap gas which is visible as an air bubble. Right? So I'm going to place this into my incubation location for 12 hours and observe color change. If there is no color change, I'm going to do an additional 6 hours. And I'm going to be looking for these colors. Okay. So this is the carbohydrate fermentation testing results. So this is fermenter with gas production, and non-fermenter. Alright, and that concludes this lab. Bye!